In pharmacology, for a drug to exert its effect on cells, it must bind to receptors located on the cell surface or within the cell. These receptors function as channels for signal transmission triggered by the drug, causing changes in the receptor's chemical structure, which leads to a cascade of reactions resulting in a specific biological response. Types of receptors. 1. Ion channels. Examples include acetylcholine receptors found in skeletal muscles, which control the passage of sodium ions. When acetylcholine binds to the receptor, it opens the channel to allow sodium ions to enter, causing a change in membrane potential and triggering an action potential, which ultimately leads to muscle contraction. 2. Second messengers. Many receptors can initiate a series of intracellular events through what is known as second messengers, such as G-protein molecules, which act as intermediaries between the receptors and the cell membrane. Second messengers activate various enzymes within the cell, such as adenylate cyclase, which converts ADP into CAMP, cyclic adenosine monophosphate. CAMP, in turn, activates specific proteins through phosphorylation, influencing various cellular functions. Key terms. Agonist, chemical substances that bind to receptors and trigger a specific response within the cell. For instance, acetylcholine acts as an agonist by binding to muscle receptors and causing contraction. Antagonist, substances that bind to receptors but prevent the agonist's effect. An example is the drug D-tubocurarine, which blocks acetylcholine from binding to muscle receptors, leading to muscle relaxation. Mechanism of drug action. In general, a drug works as follows. 1. Binds to a cell receptor. 2. Causes a change in the shape or charge of the receptor. 3. Opens ion channels or activates a series of enzymes and second messengers. 4. Produces a specific response, such as muscle contraction or various secretions. This explains that a drug's effect relies on the receptors it interacts with and the second messengers it activates within the cell.